www.nanohub.org.
You can follow along with this presentation using printed slides from the NanoHub. Visit www.nanohub.org and download the PDF file containing the slides for this presentation. Print them out and turn each page when you hear the following sound. Enjoy the show.
Okay, hello. Today I will be providing you an introduction to Biochip, Biosensors, BioMEMS and Bionanotechnology. My name is Rashid Bashir. I'm at Purdue University, the School of Electrical and Computer Engineering and School of Biomedical Engineering. So as the title says, I will really try to provide you in these following 3 lectures an introduction to a broad area of research that is now in widespread in most universities in the US and across the world in the area of biochip, biosensors, BioMEMS and Bionanotechnology. Also, I should let you know that these notes are going to be available on the web for you to download. Okay, here is an outline of the key topics that I will go through. I'll give you some basic introductions and some definitions of biochips and sensors and talk about device fabrication. Then I'd like to provide a very brief fundamental background and introduction to cells, DNA and protein, sort of essentials of micro and cell biology. And this would cover the first lecture. Then in the second lecture we'll talk about microfluidics and some biochip sensors and detection method, and this will probably carry over to the third lecture. We'll finish that and then talk about microarrays and some examples of lab-on-a-chip devices. So some basic definition, BioMEMS stands for biomedical or biological microelectromechanical systems, and these are biomedical or biological applications of these MEMS devices, and MEMS again stands for microelectromechanical systems. Bionanotechnology can be categorized as the biological applications of nanotechnology, where we all know that nanotechnology is the science and technology of miniaturization at scales of less than 100 nanometers. So how I like to sort of categorize this entire area of BioMEMS and Bionanotechnology is by the following figure shown in the slide. The goal really here overall is to apply micro nanotechnologies to develop novel devices and systems that have a biomedical impact or are bio-inspired. So if you look at the circle in the middle, you're talking about micro nanotechnology in systems and biology and medicine. These things on the left are really how we can learn and apply micro nanotechnology in systems to solve important problems in biology and medicine. And resultant areas include diagnostics, things like biochips, quantum dots, silicon nanowires, carbon nanotubes, therapeutics, which includes devices and mechanisms for drug release and targeted delivery, and also hybrid biodevices. So these things on the left are really going to provide novel solutions for the frontiers in medicine and biology, and things on the right is sort of areas that represent what can we learn from biology and medicine and how can we apply them to solve important problems in micro nanotechnology and systems. So areas like self-assembly that can be DNA or protein mediated, or self-assembled monolayers, the entire area of molecular electronics, especially the devices that use biological molecules like DNA, and things like bio-inspired materials. As an example, human skin is really a great material. It self-heals and repairs, and it senses and responds. So, if we could develop materials that mimic biological functions, then that would really be very powerful. So these things on the right will provide novel solutions for frontiers in materials and information processing. Okay, this slide just really shows you an idea of the size and scale of various objects that can be found around us. These things on the left side of the scale, so the scale actually goes from 0.1 nanometers all the way to 100 micrometers, and 1 micrometer, as we all know, is 1/1,000,000th of a meter, 10 to the minus 6 meters, and a human hair, as an example, is about 100 micrometers in diameter. So these things on the left actually are biological. They have been fabricated bottoms up, biological or chemical. most plant and animal cells actually are in the order of 10 to 100 micron. Most bacteria are in the order of 1 to 10 micron range, typically a few microns. This is a picture of E. coli. Viruses are smaller than that. They're in the order of 100 nanometer or less, something in that range. And then you have proteins. You can talk about 1 turn of a DNA, which is about 20 angstroms, about 2 nanometers, and so on and so forth. Things on the right here in the middle really are these top-down structures. So these are structures that have been fabricated using top-down techniques, which means that we have fabricated them from a larger structure and made them smaller and smaller. So most microelectronic and MEMS structures, might be in the range of 1 to 100 micron range. The minimum feature of a MOS transistor in the year 2000 is actually a little bit below 100 nanometer. This is in commercial production. This is a picture of a polysilicon gate with nitride spacers. And then one of the gate insulators, which is one of the most critical elements of a MOSFET for these 100 nanometer devices, is something in the order of 2 to 3 nanometers. So we can today make these structures that are ranging anywhere from 100 microns down to a few nanometers with great precision and accuracy. And this provides us an opportunity to really merge these two worlds, the biological world and this non-biological world, to make integrated sensors and devices. And one example of these kind of devices would be these integrated biochips, which contains macro, micro, and nanoscale structures. One example of such a device would be what's shown here, which is a 2D CMOS platform with molecular devices in memory, microfluidics, molecule-specific sensors, and MEMS and NEMS, for example. So a few more definitions. So we're going to talk a little bit more about biological sensing in these lectures. So I will just define biosensors for you, and biosensors are defined as analytical devices that combine a biologically sensitive element with a physical or chemical transducer to selectively and quantitatively detect the presence of specific compounds. And similarly, biochips can actually be defined as what I would call microelectronic inspired devices that are used for delivery, processing, analysis, or detection of biological molecules and species. And really these devices have been reported to be used to detect cells, anything ranging from cells, microorganisms like bacteria, viruses, proteins, DNA, nucleic acid, and other small molecules. So really biochips is a very broad word. It could include macro-scale structures, micro-scale structures, or nanoscale structures, and that are really used to deliver, process, analyze, or detect anything that's biological. So an overview of a biosensor system would be something along this line shown here on this slide, where on the very left you have a starting sample. The sample could be water, food, air, or some bodily fluids. You might want to do some processing and concentration steps to extract what you're looking for and to eliminate what you don't want to detect. And then you would have your actual sensor and perform your detection and identification and then provide the results. So all of this would be the components of a biosensor system, and the detection ID, this detection and this particular step right here is really what's performed in the biochips. There's also more and more reports of performing processing and separation of samples also on a chip. So let me talk about biochips a little bit more and provide some key attributes of these devices. So there's 4 key elements of these biochips. The first one is they have small length scales. So you're talking about very small dimensions, things in the order of a few microns to tens of microns. Because of the small size and small length, there's a small thermal mass. So that's an important property for some applications. At these scales, the fluid flow properties are unique, and more specifically there's a parameter called Reynolds number, and this Reynolds number ends up being about 1 or certainly less than 1 at these dimensions, and hence you get laminar flow. So it's actually pretty difficult to get turbulent or mixing turbulent flow or mixing in these devices. You have to actually take some special effort and go the extra mile to try to get mixing and turbulence. Usually it happens only by diffusion, because the fact that the Reynolds number is less than 1 and you really get laminar flow. And the last attribute of biochips is the fact that they have a high surface area to volume ratio. So if you are talking about attaching proteins to surfaces and trying to minimize the volume in which that's happening, then biochips would be very, very useful. What's shown here on the right is actually this classic picture from Whiteside's group at Harvard, which shows different flow lines. This is a solution with different dye injected into a device, and then you can see that the flow is so laminar that you can actually separate these stream lines into these different outlets, and the flows don't really mix with each other because the flow is very laminar. So some of the reasons for miniaturization include the following. So the use of micro and nanoscale detection methods is really justified by the following reasons. First of all, if you reduce a sensor element to the size and scale of the target species, then that will provide you with higher sensitivity. And conceptually, that makes sense, and that's pretty straightforward. So you can try to get to single entity or single molecule detection. So if your sensor element is as small as your smallest unit that you're trying to detect, then that can provide higher sensitivity, or single molecule sensitivity, or single entity sensitivity. Because the volumes are smaller, you also have reduced reagent volumes and associated cost, and hence each test can be much cheaper than the associated macro scale tests. Reduced time to result can also be a big advantage due to small volumes resulting in higher effective concentrations. Of course, if the system is small, the entire lab or the assay is on a chip, and hence the term, by the way, lab on a chip, that's used. So if you have a lab on a chip, then obviously you can also make it more portable, and the rest of the system can also potentially be miniaturized. You can perform point-of-care diagnostic if your sensors are small and your devices that are doing the detection are small. You also have the potential of using multi-agent detection capability if you form arrays, and we'll talk about that later in the lecture. And there's a potential for use in vitro as well as in vivo, which means in vitro meaning outside the body, or in vivo meaning inside the body. So there's lots of applications for these biochips. The applications include medicine, human diagnostics, clinical diagnostics, pharmaceuticals. This would be drug discovery and pharmaceutical applications, food safety and security, and also homeland security. For these sensors, you want the devices to be integrated, sensitive, provide rapid result, and also you have to keep in mind the cost-performance product. And there's been some examples of commercialized biochips already. Companies like Nanogen and Affymetrix were some of the first to commercialize DNA microarrays, which are really an example of biochips. And then other companies also like Caliper and Cepheid and many others have subsequently commercialized these biochips. I should point out that almost all examples of commercialized biochips now are in the area of DNA microarray and DNA detection. So these sensors, these microscale and nanoscale sensors and biochips can also be used as novel tools for what we would call nanobiology, or really understanding single cell biology. So if you look at a cell, and we'll also talk about this a little bit later, the cell is performing a variety of very complex functions. The message and the information in the DNA is converted into mRNA through a process called transcription. mRNA, then the message is actually converted from mRNA and actually translated through a process called translation to proteins, and proteins are formed, and these proteins actually are the ones that actually in turn control these processes of translation and transcription. So this is just one little example that just basically says that if you can try to make sensors that can probe single cell, analysis of single cell, and study of their function in real time, then that would be useful in understanding these processes of transcription and translation. These kind of sensors could provide increased understanding of signaling pathways inside the cell. This is a challenging problem, but certainly a goal of many research groups currently. Basic cell functions such as differentiation, reproduction, apoptosis, etc., and their implication on various disease states could be determined, or you could get more insight using these micro nanoscale sensors. And understanding the single cells and their function is really a focus of the post-genomic and systems biology era. And I believe, and many people believe, that these micro and nanoscale sensors will play a key role in this area of research. So let me briefly talk about the materials that are used to make these sensors and these systems. So materials that are used to make biochips or BioMEMS. So certainly silicon and the microelectronics material that are used to make standard MEMS are all included. I mean, we do use silicon devices and related materials like oxide, nitride, polysilicon to make these biochips and sensors. In addition to silicon and related materials, glass and quartz are also used very frequently. For example, glass and quartz are used to usually bond or cover a chip. If you're making a microfluidic device, you could cover it with glass, and that provides a transparent material through which you can actually shine light and look for fluorescence, for example. Various types of polymers are also being used to fabricate biochips and BioMEMS devices. Things like PDMS, polydimethylsiloxane, is probably one of the most commonly used polymers to make microfluidic devices. PMMA is another polymer that's been used in materials like Teflon. And in addition, we talk about all these materials like silicon, glass, and polymers, and now more and more people are talking about actually the use of biological entities to make devices. And this is really sort of a very new area of research, but can we actually use biological entities themselves, like cells and proteins and DNA, to actually make certain shapes and structures and devices? This is really one of the frontier areas of research in BioMEMS. So things like inkjet printing of cells and proteins and DNA, and things like growing cells on three-dimensional scaffolds or into three-dimensional scaffolds for hybrid biodevices are very interesting area of research, where you can think of this as an area of fabrication, except that the fabrication materials are themselves biological entities. Okay, so let me give you a brief introduction to device fabrication. In the area of MEMS and NEMS silicon fabrication, these processes are used for the formation of structures that can be used as sensors and actuators. And this is really sort of the standard area of MEMS. In this particular case, you're processing electrical and non-electrical signals, like mechanical signals, for example. So you can process mechanical signals. Conventional and new semiconductor processing technology modules are used, such as etching, deposition, photolithography, oxidation, epitaxy, and some specialized MEMS modules like deep RI, thick plating, etc. Typically, you divide up this MEMS fabrication into two areas called bulk micromachining and surface micromachining. Bulk micromachining really means that you are doing some processing in the bulk of the silicon, and it's mostly subtractive, meaning that you are actually etching something out. For example, right here it's a cross-section of a silicon wafer, and you can actually open up a window and etch this region out and create a little well in silicon. Or you can use a deep RIE to actually bombard ions down with high energy and actually form this deep trench. Or you can form a thin diaphragm of P+ silicon and then etch the backside completely to release this diaphragm. So these are examples of what people usually call bulk micromachining. And then there's examples here of surface micromachining, which are mostly additive processes, where you are actually talking about adding things to the surface. So here, for example, you can deposit a layer of polysilicon on a sacrificial layer here, which was then subsequently removed. So this layer actually right here was first deposited and then subsequently removed to form a thin suspended membrane. You can plate metal on top of it, and so on and so forth. So these are sort of additive processes which are done on a surface, and hence it's called surface micromachining. And some examples of MEMS include things like shown here. This is a bulk micromachine accelerometer, which has this proof mass and piezo resistors in this region, and this device can actually detect acceleration. Here is the probe tip for an AFM, one of the cornerstones of nanotechnology, where this probe right here, this tip is atomically sharp, and it can be made of silicon or silicon nitride. This example right here is one of the DMD digital micrometer display chip from TI that's now found in most commercial projectors. It is composed of a chip here, where the chip actually has lots of these metal mirrors, essentially, where this mirror can be deflected along an axis, and it can send light towards the lens or off the lens, and hence projected. So essentially your image is now being sent digitally to the chip, and each little mirror is addressed to turn the pixel on and off. More examples of these sensors are things like the silicon chip accelerometer, single chip accelerometer from analog devices that's in commercial production and is found in many cars now, where the deployment of the airbag is actually performed by these chips. So there's a sensor here in the chip that detects deceleration and sends us the appropriate signals to actually deploy the airbag. Here's a picture of a silicon microphone, which can be used as an acoustic transducer, or essentially a microphone on a chip, and hence you can form arrays of this for different applications. So examples of BioMEMS devices now are the following. So again, similar techniques of fabrication have been used to make sensors and probes and arrays for biological or biomedical application. On the left here you see a probe. This is actually a neural probe which has these electrodes patterned on, and this is inserted into a brain tissue to monitor electrical activity. Here is another example of a neuroprobe also, where these probes are very sharp and long, and they have these electrode regions to actually measure the electrical activity of neural tissue. Here is another example of neurons that are growing on an array of MOSFETs. This right here is a nice example of a drug delivery system. This is a microneedle that's been microfabricated, and this is a human skin with hair. So here the basic idea is that the needle of a mosquito is so small that when it bites you, you don't really feel it, and the reason is because the needle is smaller than the size of your pain receptors. So if we can micro machine such a needle, then this can be a drug delivery system which can be part of a patch that can release specific drugs to the patient without really any pain to the patient. And there's a company that's been trying to commercialize this. This is an example of a microfluidic device that can take in a sample of fluid and rapidly concentrate the bacteria that's present in it, in that sample, and then also detect the presence of live bacteria very rapidly. This is a very nice example, a classic example of these micromechanical sensors where you have a cantilever array, and these cantilevers are essentially diving boards in a swimming pool. You can think of it in that manner. And these structures here can actually vibrate or bend, and when biomolecular reactions take place on the surface, then that results in bending, and you have a label-free means to detect a biomolecular interaction. So again, for the BioMEMS and biochip fabrication, in addition to silicon, we also have additional criteria to be kept in mind. For many applications, biocompatibility is very important, and that's needed for biomedical devices. You also want the devices to be transparent within the visible range for optical detection. You would like to be able to do rapid fabrication if you can, just to be able to have different versions of the devices, and also because the fact that eventually many of these devices will be used as a one-time use device, hence the fabrication should be cheap and rapid. You want the materials to be photodefinable and also chemically modifiable, to be able to attach specific proteins or DNA or to treat the surfaces in a manner that you like. And possible examples are these PDMS, hydrogels, Teflon, and SU8, for example. So let me talk about some alternative fabrication methods that are used in this BioMEMS area, and I'll briefly go through all of these. So this area is often called soft lithography, which really comes from actually Whiteside's group, and the idea of using soft material to do lithography, such as so in this case the lithography is not really dependent or being done by light, but rather some soft material. And you can broadly categorize these non-silicon fabrication techniques into these three methods: the soft lithography, compression molding, and inkjet printing. So the first type of soft lithography is actually called replication and molding, And here the basic idea is the following, that you actually make a master mold first, and it's made from silicon, glass, metal, or let's say SU8, which is a negative resist that can be formed with actually thicknesses in the order of 25 to 200 microns. So you first make a master, and then you do a surface treatment of that master, and then once you've done that, then actually you pour your PDMS components, which is the oligomer and the cross-linking agent, and then you actually cure this for a certain amount of time. Then you would simply peel off that PDMS structure, and that PDMS structure that you peeled off basically becomes your structure that you're trying to define. And in the mold, however, can be used again and again. So that's why this is called replication and molding. And here is the picture, cross-sectional flow, and this is detailed in a review by Whitesides. So here's a silicon with a certain material as a master. PDMS is poured and cured, and then the PDMS can simply be peeled off and released, and the master can be used again. Micro contact printing is a different version of that. So here what you're actually doing is that you actually form the PDMS structure, like for example you did with replication and molding, and then once you form the PDMS structure, you could actually ink this layer of the PDMS, this surface right here, with specific molecules. For example, you can use proteins, DNA, or let's say alkyl thiol molecules. And then what you do is you actually transfer this biochemical layer through a physical contact to another substrate, and hence the name micro contact printing. So essentially you're stamping. It's like, you can think of it as stamping. And you have to optimize the time, temperature, and other properties of that step of the physical contact, and then the inking is actually performed via covalent binding, covalent bonds on the substrate. So for example, on the substrate you could have a gold layer, and then you could transfer this SAM layer from the PDMS to the gold. And this can be performed actually on flat surfaces or curved surfaces, theoretically. And the idea here is that once you have made one of these stamps, then you can keep repeating and forming these SAMs on multiple structures. Okay. One other example of using PDMS and glass to make a hybrid chip, for example, would be the following. You can actually take a siliconized silicon mold. So you have a silicon mold here that you actually silinize the surface. You form a first layer of PDMS and cure. Then you can actually take any other mechanical structure, like even a Teflon tubing, and use it as a mold actually, and pour the second layer of PDMS on it. And in this particular case, then you can actually pull this Teflon tubing out, and then even close this region right here, seal it with PDMS. You could then actually punch holes through these layer and form a vertical channel, and then attach this composite layer to another device that has channel. And then you attach tubing to these channels, and now you have a microfluidic device that's actually a composite of PDMS and, let's say, glass or silicon. And these are examples of these kind of devices that we have built. What you see here is a picture of a silicon device that has actually 3 layers of PDMS. A cross-section is shown here. There's a first layer of PDMS that has an opening, then a second layer of PDMS that actually has a channel, and then a third layer of PDMS that has another opening. And actually there is another reservoir here, essentially this large, very thick layer of PDMS. We can drop a sample and then pull the fluid through this channel out this tube, the output tube. Another example is a device here which is built on glass with some metal contacts. This device, originally the idea of air valves, actually the use of channels as air, this was originally performed by Steve Quake. And the idea here is that if you can make these channels that are within the top layer, then when you pressurize these channels with air or fluid, then these thin PDMS layer can actually bend and you can make valves. And once you make these valves, you can actually operate them sequentially to make a pump, for example. Okay, another method that also kind of falls into this area of soft lithography is what's called dip-in lithography, and this was originally performed and demonstrated by Merkin and his group up at Northwestern. And the idea here is to actually use the tip of an AFM to write molecules. So you actually have an AFM tip that's made out of silicon or silicon dioxide or silicon nitride, and you coat that with molecules. And basically there's a water meniscus around this tip, where if you were to control the relative humidity and the temperature around the tip or where this process is taking place, and you actually bring this tip down to the surface, then these molecules would actually transport down to the surface, and you could essentially write, so to speak, the molecules using an STM pen down to nanoscale, just like you would with an ink pen. And this technology is being commercialized by a company called Nano Inc. And there's been various reports of actually drawing and writing SAMs, DNA, and proteins. This process is actually typically serial, but there is efforts ongoing to actually make parallel arrays or arrays of cantilever for actually parallel writing. And you also need a continuous source of the molecule, and hence microfluidics and devices are actually being integrated into the tip to supply the fluid. Okay, then the second area is compression molding, and here essentially you are talking about the manufacturing processes that most mechanical engineers and industrial engineers are very aware of, and you use the same processes except that you actually have miniaturized it. So the process of hot embossing is actually the following. You take a mold, and this could be again made in silicon or metal or some sort of hard material that can withstand some temperature and pressure, and essentially you take a thermoplastic material, some sort of a polymer that you spin on or deposit on a substrate, and then you essentially take this mold and you press it against this thermoplastic material at specific temperatures and pressure, and you can transfer that pattern of the mold into the material and then pull the mold out. So this area of hot embossing it has been done a lot at the macro scale, and more recently it has also been done at the micro scale now. And certainly the idea of precision injection molding is something that is finding increasing use. So the process of injection molding, where you take where you make a metal mold and you inject plastic at very high pressures and at temperatures in the order of a few hundred degrees C, and this plastic will actually conform inside the mold, and then you open the mold and essentially release your plastic structure. This is the process of choice for making these plastic cartridge, for example, or any sort of plastic material that you find around you that needs to be made in large quantities. That's made by injection molding. And then these injection molding processes have been now perfected to a point where you can go down to features down to 0.1 microns and 0.6 micron wide, for example, for a CD. Those are injection molded tracks. Okay. So an extension of this hot embossing is really what's called Nano-Imprint Lithography, and here the basic idea is that if you can make your mold, your master structure down at nanometer scales, then you can transfer these patterns to the appropriate polymer at the nanoscale. And here there's pictures that show the imprint mold with 10 nanometer diameter pillars. Here there are 10 nanometer diameter holes that are imprinted into PMMA, which is an e-beam resist. And here, using that pattern, then 10 nanometer diameter metal holes have been defined using liftoff. So this is really a nanoscale extension of hot embossing, and it needs a nanoscale master mold, and this process actually now has been added to the ITRS roadmap for the fabrication of the end of the roadmap devices. And there's a company that is trying to commercialize this approach. Okay, so that basically was a brief introduction on biosensors, biochips, BioMEMS, and device fabrication methods that are used to fabricate those structures. Now what I'm going to do is switch gears a little bit and give you an introduction on cell and microbiology, such that so that in future discussions you have an idea about what DNA and proteins and cells are. So let me just get right into it. This is actually a picture of a cell from a basic biology textbook that's also called a cell. The bottom line is that the genetic information in a cell is really contained in a material called chromatin, in a region called chromatin, which is shown right here, this region right here. And this chromatin is really a diffuse mass which distinguishes two chromosomes when the cell is ready to divide. And as you all might know, that humans have 46 chromosomes in each cell, except in the reproductive cells, which have 23 each. So the 23 chromosomes each from the reproductive cells join to make the 46 chromosome in all other cells of humans. And these chromosomes are long, uninterrupted, packed, supercoiled chains of strands of DNA, and DNA stands for deoxyribonucleic acid. And it's very interesting to note that the chromosome in a human cell, for example, if you could actually extend one chromosome, it will be about 6 centimeters long. So it's really super packed and coiled inside a volume that's just a few microns cubed. And in human, each chromosome is in the order of 50 to 400 times 10 to the 60 units long. So what would be very useful to know, for me to tell you, is these basic processes that take place in a cell that transfer the information from DNA to proteins. So the basic idea is that the actual information in a cell is actually static, and it's fixed in time, at least over a short period of time. Over long periods of time you can have DNA mutations, but let's say the information here is relatively fixed, and there is a process called transcription which converts the information from DNA into a molecule called a messenger RNA or mRNA. And then the mRNA then has the actual information, which is then translated into the formation of proteins. So really the formation of proteins, or the synthesis of proteins, is the essential step that, or the essential function that keeps a cell going, and that provides all the mechanisms for the cell to continue its function. So the information, the DNA, the base pair sequence of DNA and mRNA is very, very important, because that results in the formation of specific proteins. So the process of transcription is the process where double-stranded DNA are actually converted to a single strand of mRNA. Okay? So the idea is that you have a DNA is actually a double-stranded structure. It has this double helical structure, and you can think of the DNA structure to be similar to a twisted ladder. So you take a ladder and you twist it, and that's basically the structure of a DNA. And basically the two ends of the ladder are connected by these base pairs. So you have base pairs that connect the two backbones of the DNA. So there is a molecule called the RNA polymerase, and this molecule actually synthesizes the messenger RNA. Okay. And this is schematically shown here on the right, where you have a double-stranded DNA. And the DNA transcription, actually, what this process does is it actually separates these two strands and reads the information and then creates a replica of it, which is called the RNA. And it turns out that this messenger RNA is actually a condensed form of the information. There's actually more bases here in the DNA than the information that's in the RNA. But what we are really interested in is this information in the RNA. And then what happens from the RNA is that there's a process called translation, where these little nanomachines called ribosomes actually go and read the sequence of bases in the mRNA. And each 3 bases are called a codon. So each a triplet, each 3 base is called a codon, and each codon translates to a specific amino acid. And these amino acids then are connected together to form a protein. So this is what the ribosome does. It actually reads the sequence of these codons and then picks out from the medium around it, from the solution or chemicals around it, it picks the appropriate amino acids and connects them to form the protein. Okay. So this is just another picture with decreasing complexity of a chromosome. A chromosome are these sort of double Y-shaped structures. This is a drawn picture and this is an SEM picture, or rather a TEM picture. And when you try to look and zoom in into this into the chromosome, you will eventually see the DNA double helix that we're all used to seeing. So let's talk about DNA a little bit more. DNA is composed of actually a sugar phosphate backbone. So this backbone of the DNA molecule has a phosphate group which has a net negative charge on each phosphate. So you have this PO4 minus group, okay? So there is a negative charge on each phosphate, and then this phosphate is connected to a sugar, okay? And this basically, this sugar is called deoxyribose, and that's the D in the DNA. So it's a deoxyribose nucleic acid. So you have a sugar phosphate backbone, and these two molecules form this a chain of these two molecules form the backbone, and then to each deoxyribose, to each one of these sugar, then you have a base attached. And these bases, we actually have 4 types of bases or nucleotides, 4 bases or nucleotides, and these are called A, T, C, and G. A is for adenine, T for thymine, C for cytosine, and G for guanine. And so what you also need to know is that these bases are complementary bases, which means they are selective to each other. A specifically binds to T only, and G binds to C. So A cannot bind to G and T cannot bind to C. Okay, so A binds to T and G binds to C. So what happens is that you have 2 of these strands, and this line right here basically just shows that sugar phosphate backbone schematically. And here are your bases. So this is your T and this is the A. And A only binds to T, and the reason it only binds to T is because actually the bonding is through 2 hydrogen bonds, okay? Whereas G and C bind to each other with 3 hydrogen bonds. So because of this number of available binding sites, or these hydrogen bonds, you cannot have 3 bonds bind to these 2, because that will not be the lowest energy state. So A binds to T and G binds to C, and hence these 2 strands come together in a complementary fashion. Some other properties about DNA that are very useful to know. DNA hybridization is a process where actually the 2 strands come together and bind. So now if you take a double-stranded DNA and you heat it to a temperature that is, let's say, above 90 degrees C or exposed to very high pH, then the complementary strands actually can dissociate, and that process is called denaturation. So essentially you can break the hydrogen bonds between the bases. This process is reversible, and actually exposure to, for example, a melting temperature of about 65 degrees or so, and 2 complementary single-strand DNA will hybridize to each other and form a double strand. So what you can do is you can give them just about enough energy to break the bonds, and then when you lower the temperature again, then those 2 complementary strands will actually come together and form a double strand. Hybridization can take place between any 2 complementary single-strand molecules, DNA to DNA, DNA to RNA, RNA to RNA. Okay? And hence it can also provide a very sensitive means to detect specific nucleotide sequences, so short sequences. There's lots of factors that affect hybridization, this process of the 2 strands coming together. That's called hybridization. And there's a lot of factors, including temperature, of course, salt and buffer concentrations. This is very important, salt and buffer, because that can change the net ionization of the molecule and of the phosphate backbone. So the charge on the phosphate backbone could be neutralized depending on the salt and buffer conditions. The GC content, and by the way, using these conditions, you can actually calculate the melting temperature of a given sequence of molecules. There are formulas available to do that. And of course, the rate of hybridization is proportional to the concentration of target and the probe, and limited by the lower concentration materials. So this is what you might have. You have the native DNA, which is a double-strand structure, shown here as a ladder, but it's actually twisted. And if you heat it, then these 2 strands will come together. And if you then cool it down slowly, then these 2 strands will actually come together. And if you give them still some thermal energy, then they will come together and actually bind again. Okay. And what you measure here is the relative absorbance at 260 nanometers as a function of temperature, and you can get this melting curve for a native DNA, for renatured DNA, and for actually a denatured molecule. Okay, so this, you also need to know this concept of stringency, which means that actually depending on the number of bases that a molecule has, number of bases that a double strand has, you can actually still have a case where you might have a few mismatches here. For example, this is a mismatch, this is a mismatch, but the two strands might still come together. So for example, if you have a few mismatches in a base in a molecule that's 20 to 30 bases long, the two strands will still come together, but it's called a reduced stringency hybridization. A perfectly stringent hybridization would be one that's perfectly complementary. So it's important to note that you can have mismatches and the two strands will still bind. So I want to briefly talk about this technique called PCR, which stands for polymerase chain reaction, and this was a technique to amplify or to make multiple copies of known DNA molecules. This is a very important, very important process, and that really revolutionized the area of biotechnology, invented in '85 and subsequently commercialized. And this is basically an amplification process for DNA strands. You cannot do amplification on proteins to date, but you can do it on DNA. Okay, so what you do is you actually use these enzymes called DNA polymerase and primers, which are short single-strand DNA strands. So there's an enzyme called DNA polymerase. You use that, and then you use molecules called primers, where primers are simply short single-stranded DNA strands. Using these techniques of PCR, you can make billions of copies within a few hours, and it's been very useful in research, diagnostics, and forensics, where large samples are required. So you might take a small sample from someplace, and then if you know what DNA strands you're looking for, you can try to amplify that to a very large number. And the way it works is the following. So let's say you have a double-stranded molecule shown here. What you would do is you would actually heat up this molecule to a temperature at which it denatures, which means the two strands separate. So that might be, let's say, 90 degrees or so. And you do that for a very short time. Then during that time, you introduce these short molecules called primers, and now here is a trick. This is one of the tricks. You have to know the sequence of these short complementary regions. So these primers are in the order of 5 to 10 bases long, and you design them yourself. So you have to know at least, or take a pretty good guess at this initial regions of the molecule which you want amplified. So these primers would then bind to these two strands, and then what's going to happen is that you actually perform the DNA synthesis using this enzyme called the DNA polymerase. Okay, so DNA polymerase is an enzyme which takes nucleotides from the ambient solution and starts to construct the complementary sequence. And then you let it make the complementary sequence, which happens in this step. So you actually have this denature step, then you have a lower temperature anneal step at which you anneal or react the primers to the main strand. Then you do an extension step, and this is where the DNA polymerase is working and actually extending the primer. That's why it's called the extension step or the extend step. And then now you have formed two copies of that double strand. And you again do a denature step, which means that these two now are going to separate. And then you again go down to an anneal step where the primers bind and anneal to the long strand. You do an extension where the DNA polymerase extends it and makes the copy, and then you continue this process. So you can run one cycle in just about a minute and a half or so, and essentially this is an exponential process. It doubles every couple of minutes, so you can make a large number of copies in a very short time. You need an adequate supply of the nucleotides, which are the individuals A, T, Gs, and Cs, because those are needed by the polymerase, by the DNA polymerase, to actually form the complementary molecule. Okay, so proteins then, let me talk about the protein structure. Protein are actually complicated three-dimensional structures that this is one unit of a protein. You have this NH2 group, carbon in the middle, and a COOH group here, and then you can have a different side chain, essentially a side group. And depending on these different site groups, you actually have different units of this protein. So one of this would be called an amino acid, and depending on the different side chain, you have different amino acids. And the different amino acids then are combined together, are bonded together by a peptide bond to form a protein. Okay? Okay, I think we'll stop right here and continue the rest of this in the next lecture.